everyone, this is Marissa. I'm going to present my gummy bear experiment for the first interactive lab that we have this semester. For step 1 of the experiment, I filled 2 cups with tap water from my sink. In the first cup, I kept it just pure tap water and I didn't put any salt in it as suggested. And then in the second cup, I just kept adding salt and stirring and repeating this process until the water could not dissolve any more salt into it. And I made a saturated salt solution by doing this. In step 2 of the lab, I chose 2 red gummy bears because I thought it would be best to keep the gummy bear variable constant and it would be easier to see the change if they were the same color. I placed 1 of each of these gummy bears in each of the cups and I set a timer to ensure that I would wait 8 to 10 hours before taking them out of the cups. Here is the results that I got from doing the experiment. On the left is just a gummy bear that I got from the package. I put it there just to be able to compare it for size to the other 2. The gummy bear in the middle is the one that I got from the water, just the straight tap water, and it has obviously swelled up. It is much bigger than the normal one and the salt gummy bear that's on the right. The gummy bear on the right is the one that I had put in the in the saltwater solution, and it looks like it has actually gotten a bit smaller than the original gummy bear, so that's interesting. A hypotonic solution could be the reason why the gummy bear in the normal water actually swelled up, and this is because there was an unequal concentration gradient. So basically what this means is that the gummy bear has more sugar molecules than the water does on the outside. So the water will flow into the gummy bear to make sure that there's an equal concentration gradient between the two, between the cell and between inside the cell and outside of the cell. So once this concentration gradient is equal, then no more water. The water in the cell and out of the cell will be equal. A hypertonic solution could explain how the gummy bear put into the salt water shrunk a little bit because the salt water has much more salt molecules on the outside of the cell than there is inside of the gummy bear cell. So when there is more salt on the outside, this means that there's a higher concentration than the interior. So the solvent will flow outside of the cell to try and get more water out into it to make it equal. And this causes the gummy bear to shrink. And this is an isotonic solution. So an isotonic solution, so if the solvent, which is in this case either the tap water or the salt water, had the same concentration as the cell, so the gummy bear, then the solvent molecules will transfer in and out of the cell at the same rate, so the size of the cell would remain the same because nothing is going in or out of it unequally. The concentration gradient would be equal. And this pretty much sums up what I found out from the lab. It was actually successful. Everything that was supposed to happen happened, so yeah, it went well. That's my presentation.